Thank you so much, Jonathan Fisher. Presumably you can hear my lovely voice, and I'm going to go ahead and share a screen now on which I'll place that wonderful slide that you just saw to begin this wonderful talk. And in fact, it begins long, long ago when I was much younger. I had a lot of hair back then, and I always love giving these STEM talks to students. Here I am as a 18-year-old in my first college experience, and I engineered my first optical imaging system out of just a few lenses and a laser and a photo detector circuit that I had built by hand. And I'm starting here because I want to build to the sharing of data like we do with Snorkle and with other tools. But I want to sort of give you an idea of how someone makes this journey and where it starts. It just starts from curiosity. So by pulling and melting a glass pipette till it was thinner than a single hair, we made this cantilever that works like a springboard, right? And when we attached a single muscle fiber to it, we could use a laser to image that cantilever and measure how far it pulled when the muscle pulled. So we were able to measure the force exerted by a single human muscle fiber. And that's basic science, where you work to understand some basic principles of biology. But there's another kind of science called translational science. And translational science is the notion that we can develop methods of science for treatment of disease. And we can study this in a research hospital. So, the original mission here of the Rockefeller is shown to you, and it's primarily to use our understanding to figure out how to use science to help people. And so our subgenre is going to be light. We're going to use light to help people. Light is an amazing thing because it's both really useful and also really interesting. The biggest lobe of our brain, our occipital lobe, is dedicated to vision. And so we use light all the time to see, but what is light? Is it a wave or a particle? Why can it only move at one speed? Those are great questions. But in engineering, we use it if we can understand how it behaves. And there is a set of equations that we follow, a relatively simple set of equations that completely characterize the behavior of light. And we have fun doing it. This is Dr. David Huang, who like myself is a engineer that engineers these laser technologies. Here we're using one of his laser technologies to image the cells in my tongue. And pretty soon we'll show some cells that we can image in vivo, imaging cells without cutting anything out. But he just won this Lasker Prize, which is a big deal. It's sort of like the VMAs or the MTV VMA of science. So he can get famous doing this sort of thing too. But whether it's basic science or translational science, it's all optics, the science of light. And like all other sciences, we could not do it if it weren't for the scientists before us. So I'm going to take you on a little historical tour. This was the first person to use a microscope to look at biology. And this person wasn't interested in the microscope per se, but he was the first one to think about how a laser might work. Well, then this person took Einstein's basic theory, his basic science, and translated it into a real working laser. And he made it real. And then right about that same time, this person made a 3D microscope, a confocal microscope. We're getting really close to unlocking this great knowledge and powerful insights in biology and medicine, but it wasn't until this person figured out how to put this polygon mirror inside the confocal microscope to bounce the lasers off, make it 1,000 times faster so we can actually image living cells in human skin in real time. And that's what you see here. You see all these dark holes are single cells in the skin called keratinocytes, and you can see the little blood cells moving in. We'll see more about that later. But then I came along and I threw away all those expensive moving components, and we made the first solid state confocal microscope. And that brings us to images like this, where we can see all of these cells within surgical specimens, and we can do it in whole tissue without making a microscope slide. This is how the scanning laser microscope works. And it gives us these images where we can see cellular morphology. As a disclaimer, I have a financial interest in this technology, which has not been reviewed by the FDA, has not been proven safe or effective, and may not be used to manage patient care yet. But this is very exciting that we can do this imaging, and we want to use this technology to bring pathology to the world. I'm going to talk to you today about a different type of 3D imaging, and I'm going to show you this data. And until wonderful 3D apps like Jonathan Fisher Snorkle, we had to use this anaglyphic 3D. This was the, I don't have my anaglyphic glasses handy, but this is the old school 3D. You look at this, this looks like the city skyline of New York, and this is a melanoma. It's a very rough. Whereas this is a nevus, and it's very smooth. And this is all of 3D structure that we can extract from the confocal microscope. And so I'm going to tell you in a little bit about how that my mentor's invention with the disco ball inside the confocal enables us to do this. But first, I want to show you a few other confocal imaging confocal images. This is that confocal image on a beating heart of an embryo. And we can see in the egg with that confocal microscope I talked about, and the holes in the middle of the beating heart let a certain number of blood cells through. You can actually see them going through the holes. And if that number doesn't increase during development, there can be birth defects. So the confocal microscope can be this great instrument to study a wide range of things, not just detection of cancer or deadly disease, but the studying of how a heart develops in an embryo, in this case, a chicken embryo, in an egg where you can see. Well, another thing we can do is study human neurons. And in this example, I 3D printed and imaged a human brain cells. So I 3D printed a structure that contained human brain cells. And then we're watching as the signals propagate through. See, it looks sort of like a Christmas tree with lights on it. But each one of those lights is it's a little cell depolarizing and showing us where the signals are transferring in this neural network. So really, when it comes to imaging, first we see this image, then we sort of believe that there's some scientific or medical reality underlying it, and we use these quantitative tools to measure it, and to quantify it. And that's what we call imaging biomarkers. So what I'm going to do now is share how we can use Snorkle to visualize these things. So I just stopped sharing my screen, and I'm going to now demonstrate how we can use a different visualization tool, Snorkle, to explore data. For the young engineers out there, at one point I was creating these microscopes, and I needed to find a specimen to image with the microscopes. And the specimen that I had most handy was my skin. And so I'm going to talk about skin and show you some images. This is a close-up picture of your skin. So now that we've got that through the little snafu, I'm showing you skin here. So what you see is hairs coming out of your skin. They go down to a hair bulb. What I want to point out is that the skin is a complete organ. It has all the layers up here of the epidermis that we're going to see in the data. It has nerves, it has vessels, and it's all right there within a superficial millimeter of your body. So this is why I got into skin imaging, because I was creating these microscopes in grad school. I thought, well, yeah, let me put my finger on that. Oh, and all of a sudden you can see your own cells, which is kind of cool. And then as I grew as a scientist, I started taking this type of image, and this image is a cancer, and these are cells in a cancer that you can see with this kind of microscopy. Now, the contrast we have, the magenta is the collagen background. That's what your skin is made of, makes your skin tough. But the green, these are the actual strands of infiltrative basal cell carcinoma. So that's a non-melanoma skin cancer. And because we can get these images of this cancer non-destructively, we don't have to do something called the histopathology lab. Now, I should mention that this is not proven yet. So when I say we don't have to do it, yes, we absolutely still have to do it when we're taking care of people. But the hope is that we can take care of people better using this scanning laser technology. To give you an idea of what the device with the disco ball in it that my mentor created long ago shows, I want to show you this. And now what we're going to do is we're going to use the Snorkle viewer to go into the skin from the outside. So this is the surface of the skin, and this sort of structure you see is called stratum corneum, and it is the superficial most layer of the skin, the surface layer where all the dead cells are protecting you from, UV radiation, etc. But when you go in, by etc., I mean like infection and all that stuff, it's a barrier. When you go in, you see these weird looking cells. This cell right here, is a melanoma cell. Now that's important to detect that kind of cell non-invasively, because then we know how to where to place a biopsy to cut melanomas out of people. I don't know how familiar everybody is with cancer, but it's a deadly disease. It's the second leading cause of death in the United States. And with this microscope, this happens to be a particular confocal microscope, the Vivascope, that has an FDA clearance. You can go into the skin. So again, this is the surface. This bright circle you see is the imaging window. It's where the curved focal plane, which is a sphere, intersects an imaging window, which is a plane, and that makes a circle. And as we go in, we can see the stratum corneum, that superficial layer. And then we get in a little deeper. We see this. And this is a pattern, a honeycomb pattern you can see, where the normal keratinocytes have dark nuclei. We can see these cells non-invasively, and we can suction through them with the Snorkle viewer. Now we go deeper. I wish I had the hand raiser. Does anybody want to guess what these bright cells right here are? Melanoma cells. Melanoma cells. So because we can visualize this cell from non-invasively, we can find a very early melanoma when it's just, a few cells or a few hundred cells big, and then we can cut it right out, and potentially save a life with this technology, with this non-invasive imaging technology. Here's another melanoma. Here we're going to go in. And as we go in, oh, there are those melanoma cells. There they are. Boom, right here. Boom, right here. We could even focus a laser onto one of those cells and kill it if we wanted to. But surgical cure is the best cure for skin cancer. So, well, we saw those melanoma cells. Let's see what a nevus looks like. This one is a nevus. So when we go into it, we don't see those same cells. We see very regular pigmented basal keratinocytes. And so this is my own arm. And this is how I got into imaging, is I was looking at these cells in my arm. And so I just wanted to show you that you can see the 3D view with this viewer on these confocal images. A little bit about the shaking back and forth. People are always moving. As I was taking this image, I was also breathing, and that leads to the emotion artifact. But you can see these wonderful, wonderful cells here in reflectance mode, and you don't even have to take a piece of my skin off to do that with the kind of imaging we do, which is confocal imaging.